They still, to this day, talk about what they do is completely voluntary, but the reality is something very different, because in 1996, with the Uruguay round of the General Agreement on Tariffs and Trades, when they set up the World Trade Organization, they actually referenced, in cases of food safety, Codex Alimentarius standards, guidelines, and recommendations will now be considered to be trade legal in WTO disputes. And what that means is if there's a trade challenge between two countries, if one country is challenging another and saying that a given standard is actually not science-based but is a non-tariff trade barrier, then what WTO would first do is they would go and they would look, if it's a food safety issue, they would go and look at Codex and say, is there a standard, guideline, or recommendation? If a country meets that Codex standard, lives up to it, then it wouldn't have to worry. If it's challenged by another country, it's automatically going to win at WTO. And so the concern is that if your safety assessments are not up to standards that are permitted by Codex, then your products can get discriminated against in international markets. So there was an 8-year process, or two 4-year processes with this ad hoc task force on foods derived from modern biotechnology, and a number of documents came out. The document, which is the Codex Guideline for the Conduct of Food Safety Assessment of Foods Derived from Recombinant DNA Plants, which was finalized in 2003 at the end of this process. There were many, many fights over the language in this document. There's a whole section that's called unexpected, unintended effects that the NGOs and others were we fought very hard to get language into these sections because we were concerned that people were only looking at the traits themselves and not looking at all sorts of unintended effects. And is the case with any of these documents, what you do have is you have the different sides will get their language in. So what's here in the pink, that's the language that Health Canada points to, because one of the things they say when you talk about unintended effects is they say, well, this is something that just happens with conventional breeding or genetic engineering, so it isn't anything new or unique, and that these guidelines don't refer to stacked traits. However, as I said, Health Canada will point to that sentence that basically says that these unintended effects can occur with conventional breeding. However, the sentence that is in red was actually, this was language that Consumers International helped develop along with the European Union and others, because we were concerned about stacked trait crops that might come down the pike. And what that says is unintended effects in recombinant DNA plants may also arise through the insertion of DNA sequences. So that would be each of the genetic events. And then it goes and/or they may arise through subsequent conventional breeding of the recombinant DNA plant. And that language right there covers what are stacked traits, because stacked traits are genetically engineered varieties that are then crossed through conventional breeding. That's exactly would be the case. So that would permit a country now to require an assessment. Oh, turn it down. So in this paragraph, this talks about how you should do assessments for unintended effects. And what the industry says, and I'm sure Health Canada would say, is, well, but this is something that is done with conventional breeding, and what they do is they look at agronomic and phenotypic characteristics during the breeding process, and if everything looks fine, then they've done their job. And so therefore they managed to get this language in, again, in pink, about how the assessment takes into account agronomic/phenotypic characteristics that are typically observed by breeders in selecting new varieties for commercialization. So yes, they got that point in, but we also got the European Union, Consumers International, and actually there were other countries in Asia and Africa. As we pointed, yes, they do that, but if you look at the language that follows it, these observations by breeders provide a first screen for plants that exhibit unintended traits. Then the key language is there in red at the end. New varieties that pass this screening are subject to a safety assessment as described in sections 4 and 5. Sections 4 and 5 of that document go through the complete safety assessment that should be done for genetically engineered plants. So with the language here that is in these paragraphs, that permits a country, such as the European Union or any country in the world, they can develop national regulations that will require full safety assessments for stacked trait crops, and they can reject anything that comes from countries that do not require that assessment. And then this language that's important is that molecular, biological, and biochemical techniques can also be used to analyze potential changes at the level of gene transcription and message translation that could lead to unintended effects. That means look not just at the inserted proteins, the inserted genes, but try to look at what else you're causing to go wrong in the genome. And again, the first few sentences in paragraph 17 basically talk about you have to look at a variety of data to assess unintended effects, because no individual test can detect all possible unintended effects or identify with certainty those relevant to human health. Now I'm going to talk about a couple of studies that have found unexpected effects, and these are with simple single trait crops which had already been assessed and gone through, like in the US in 1996 and in Europe a few years later, and they've been approved here in Canada. And this is a paper that was published last year on proteomics, and that's the study of expressed proteins, and this is a good way to detect unintended effects associated with genetic engineering, particularly the disruptive effects that can happen due to the random insertion of the transgene. And this was a superior design because what this study did is they looked at a genetically engineered maize, this Monsanto's Mon 810, and they looked at the near isoline, and that means the non-engineered parent. They were grown side by side in a growth chamber, which means the environment was exactly the same. So the only differences now that you'll find in either genes that are expressed or proteins, since it's the same genetics, the only difference is the insertion of this trait, this cassette for Mon 810 with this Cry1AB protein, because your environment is exactly the same. So any differences would be the result of the engineering process. And that's what you want to do. You don't want to take plants that are not isolines or grown in different environments, because then you're going to get these confounding effects from different genetics or different environments. So this was a proper way to do it. And what did they find? They found that there were 43 proteins that were either resulted in up or down, significant up or down regulation, and that meant that either they were twice as high as the isoline, or they were less than 50%, so they were twice as low, or there could be genes that were turned off in the isoline that were turned on in the engineered in Mon 810, or vice versa. And so they said that by doing this, they could look at what were the effects that were related to the insertion of a single gene into maize. So of those 43 proteins that were different, 14 were down-regulated, lower levels, 13 were up-regulated, higher levels, 9 were shut off, that means they were on in the isoline, off in the Mon 810, and 7 were newly expressed. And as they point out, one of the newly expressed proteins corresponded to the 50 kilodalton gamazine, which is a well-known allergenic protein. So that means you had a known corn allergen turned off in the isoline, turned on in Mon 810 in this particular case. So you had an allergen being expressed, which was not there before, was also not coded for by the inserted trait. Also, they pointed out as a major concern, a number of the seed storage proteins, the globulins and viscin-like embryo storage proteins, they exhibited truncated forms. That means they were smaller, having molecular masses significantly lower than the native ones. What impact does that have on a food safety assessment? You wouldn't know unless you actually did the study. And I should point out that the seed storage proteins, they primarily are what make up the seed. So it isn't just some small level, it's those major proteins.